Okay, so thank you for joining us. My name is Scott Soderling, and I'm the CSO and founder of CasTag Biosciences. I also have a research laboratory at Duke University. So what I'm going to talk to you about today is a novel CRISPR knock-in technology for the robust analysis of cells and cells within tissues. And I think probably as all of you appreciate, the analysis of endogenous protein localization, function, and dynamics is really fundamental to the study of all cells and is integral to both academic as well as translational and industry-based research. So today, to cover this new technology, I'm going to go through several different considerations in this slide deck. So first, I'm going to tell you about this new CRISPR-Cas9 based approach called homology-independent universal genome engineering, also abbreviated as HiUGE. So I'll just refer to that as HiUGE throughout the rest of the talk, but that's what it stands for. So I'll tell you a little bit about how this new technology works and some of its advantages to other existing technologies. I'm then going to quickly cover through some of the considerations that you might want to think about and go through as you're considering a variety of different types of knock-in experiments using CRISPR-Cas9 and how these can be addressed using this new approach. I'll next then jump into examples of 7 different applications that are quite diverse as examples of how you can possibly use HiUGE to advance your own research. And then finally, we'll go through some of the validation data, talking about off-targets, efficiencies, cell types, and protein types that this new method can be used for. So most genetics research workflows start with a variety of kind of large-scale experiments to then quickly narrow in onto targets of interest. So one example of this is an active area of research that's been active for several decades now, which is the genetics of human patients that have been diagnosed with a variety of disorders who have a genetic basis. And so by high-throughput sequencing or genome-wide sequencing, mutations can be identified that are potentially pathogenic that then lead to hypotheses that can then be further tested. Another entry point for these types of experiments is omics analysis. And so omics analysis, a good example of this would be proteomics, or another example could be metabolomics. Proteomics is actually one type of analysis that I utilize quite a bit in my own research laboratory at Duke University. And we use proteomic approaches in order to identify proteins that are enriched in particular parts of neurons within brain tissue as an entry point to understanding how these neurons function to guide and modulate a wide variety of diverse behaviors. And after identifying proteins or genes of interest, either through these larger scale types of screens, often researchers then want to use a quick genetic screen, further genetic screen, to narrow down these large numbers of potential genes of interest or proteins of interest. And often this is achieved through CRISPR-based knockout screens. Once these genes of interest or proteins have been narrowed down, then another common step in most research workflows is to then further characterize these genes of interest or proteins using CRISPR knock-in technologies. And this is an area that has really advanced very quickly in the last 5 years. And really this has taken over our ability to localize proteins either in cells or in tissues, so that we can then better understand their function, and also perform functional analysis so that we can phenotype what these proteins or genes of interest are really important for, and cycle back to the entry point that we started at, which was to generate a hypothesis given our specific research question. So the last two steps, which are the CRISPR knock-in for characterization and functional analysis, are two steps that really became bottlenecks in my own research laboratory as we identified hundreds of proteins through proteomic data sets. And we really realized that we needed higher throughput technologies that allow us to do this type of CRISPR knock-in analysis and functional analysis. And this led to the development in our laboratory of a new technology that's now being marketed through CASTag Biosciences. So this is just a quick overview of how this new technology works. I'll cover some of the details of how this new technology works in just a couple of slides. In general, this new technology, or homology-independent universal genome engineering, uses a proprietary two-vector system. The first vector is the targeting vector, and this is similar to all other vectors that are used in any type of CRISPR-Cas9 based approach. So this targeting vector expresses a guide RNA that is specific for your gene of interest and guides Cas9 to that genetic loci so that it creates a double-stranded break, which is schematized by the strand of nucleic acid falling apart with a break in the middle. Once this double-stranded break has been introduced into your gene of interest exactly and precisely where you want to position it, we then have a second vector type called the payload vector. And this is actually quite unique. I'll cover this in just a couple of minutes. But these payload vectors can encode any functional recombinant DNA sequence that you would like to deliver to your gene of interest, referred to as the payload. The nice thing about this is that these payloads are agnostic to either the species or to the gene of interest. So the payload vectors that have been developed at CASTag Biosciences can be used in both, for instance, both mouse as well as human genomes. And in addition to that, individual payloads can be used across multiple different genes of interest. So it makes it very easy to utilize. And as a consequence of this, we only need to develop payloads that are specific for one of three reading frames at either the N-terminus or the C-terminus of where you would like to modify your protein. So the consequence is that with a simplified 6-vector payload system, we can essentially cover all proteins encoded in both mouse and human genomes, as well as other genomes as well. So the modularity of these payloads, because they are agnostic either to the species or gene of interest, has allowed for the creation of several payload vectors that can be used with any guide to direct their insertion into a gene of interest with different types of functions. So the first payload vector type is called EpiTag. This payload vector delivers into the gene of interest an antibody epitope tag. This is actually quite useful because a lot of times in biomedical research, we're faced with trying to understand the function or characterize further a protein of interest. However, there aren't really high-quality antibodies that exist for following up and localizing these proteins of interest. So instead of developing new antibodies based on purified protein, we essentially have flipped the process on its head, and we've taken a sequence that encodes a short amino acid sequence for which we already have an antibody that's highly antigenic and is highly validated. And so we can insert this antibody epitope tag directly into your protein of interest, and this is called EpiTag. We also have Fluor tag. This can insert larger payloads, for instance, green fluorescent protein or red fluorescent protein, so that not only can you localize your proteins, but you can also follow them in real time to image their dynamics as they exist in the cell. And then finally, the third payload vector marketed by CASTag is called DisrupTag. This encodes for an antibody epitope that is specifically designed to alter your protein function. So this can be used in two different ways, and I'll show you later in the talk examples of each of these. But for DisrupTag, you can essentially use this either for structure function analysis, to either truncate your endogenous protein of interest at different places and mark the protein containing that truncation with the antibody epitope tag, or you can place it very early on in the gene of interest to disrupt the expression of the entire protein and mark those cells that contain that disruptive insertion. So these are very useful for studying protein function. Okay, so here's some more specifics of exactly how this works. So for the targeting vector, we have a gene-specific guide RNA, which I mentioned directs the cleavage. In this case, I'm showing the cleavage at the C-terminus of your gene of interest, right before the stop codon in the open reading frame for your protein of interest. So the GS guide RNA is the gene-specific guide RNA, and we package these vectors into adeno-associated virus. And I'll talk about some of the unique features of utilizing adeno-associated viruses in the workflows in order to utilize this new technology. This then directs the cleavage of the double-stranded break in your GOI, or gene of interest. This targeting vector is then paired with a unique payload vector. This payload vector is self-cleaving. And so the way that this works is that the payload vector also expresses a donor-specific guide RNA that recognizes a donor recognition sequence. And this donor recognition sequence flanks either side of the payload of foreign DNA sequence that you would like to insert in. These donor-specific guide RNAs have been carefully chosen so that they're inert to either human or mouse genomes. So these guide RNAs don't recognize anything in either of those genomes, but instead they specifically recognize the payload vector, cleave the payload out of the vector so that it can then efficiently insert into your gene of interest. And in CASTag, we've identified the genetic rules that are required in order to utilize this, such that these payloads can insert in frame and into any gene of interest. So the upside of this is that instead of having to specify unique donor vectors for each payload and for each gene of interest, we can at large scale generate libraries of gene-specific guide RNA targeting vectors to your genes of interest, to label your proteins of interest, and then combine that with pre-made payloads. And these payloads are essentially plug and play. So in the schematic, I've got payload A, B, and C. And these payloads all can insert into diverse genes of interest once they've been unlocked and opened up by the double-stranded break encoded by the gene-specific targeting vector. This then allows very rapid and versatile modification of endogenous proteins for a wide variety of applications, including localization mapping, as well as functional manipulation. Okay, so that's a little background about this new technology, homology-independent universal genome engineering. Now I'm going to cover a couple of the important considerations that should be thought through in order to apply this new technology to your own research. So the first consideration that you should think about are the specifics of your gene of interest and the protein that it encodes. So you need to think about, you need to know something about where your protein of interest may be expressed so that you're using the appropriate cell type or tissue type in order to apply this technology. But you also need to know a little bit about something about the protein of interest. In other words, will this protein tolerate insertions or fusions either at the N-terminus or at the C-terminus? And that's really unique for each protein. Our team of scientists can also help you predict this as well if you're not exactly sure. I'm not going to cover those in detail, but I'm instead going to talk more about the CRISPR knock-in approach considerations that you should go through. And I'm going to cover the repair mechanisms, the delivery method of getting this into the cells, and then the desired payloads for your specific knock-ins. So there are several different repair mechanisms that have evolved and have been utilized for CRISPR knock-in approaches. Probably currently today, the most common repair mechanism is HDR, or homology-directed repair knock-in. This approach has some advantages as well as several disadvantages. So in general, homology-directed repair only works in dividing cells. This is not the preferred repair pathway following a double-stranded break within cells. And so the efficiency, or the percent of repairs following a double-stranded break that utilizes HDR is estimated to be somewhere around 10%. Because HDR, and I'll go through this in a little more detail in the next slide, but because HDR is using homology, this necessitates that each donor vector is unique to the gene of interest. And the insertion efficiency is typically less than 1%. It's typically somewhere around 0.1%. However, in some cases, it can be anywhere up to 5%. So the efficiencies overall of insertion are fairly low. The advantage is that when it does work, you can get scarless integration. So you can have your recombinant DNA seamlessly integrate into the DNA sequence that you're targeting. Of course, the disadvantages, though, which is a pretty large trade-off, is in the efficiency as well as the throughput, because you have to make a unique donor vector for each type. The other type of repair mechanism that occurs in cells following double-stranded breaks is non-homology end joining. And this works both in dividing cells as well as non-dividing cells, and is the preferred pathway for repair following these double-stranded breaks in cells. And so more recently, researchers have begun to realize that non-homology end joining can also be used for very efficient knock-in using CRISPR-Cas9 technology. And this is actually the repair pathway that homology-independent universal genome engineering, or HiUGE, utilizes. And so the advantage of this is because there are no homology arms, the insertion efficiency is relatively high, anywhere from 20 to 30%. It's highly it enjoys a high throughput because you don't have to design custom or boutique donor vectors for each gene of interest. It's very easy to utilize. The disadvantage, which I'll go through in the next slide, comparing in more detail HDR non-homology end joining, is that you can get both scarred in addition to the scarless integration. And so that's been considered a disadvantage, but I'll cover why probably it's not that as big of a disadvantage as previously was thought. So why use non-homology end joining rather than HDR? So I mentioned that homology-directed repair. So in this example, what I'm showing you is an example of where you might want to insert something such as green fluorescent protein into your gene of interest using homology arms on either side. Now, these homology arms, the length of these homology arms can be quite variable depending on the type of insertion that you want to do. For something that's relatively large, such as green fluorescent protein, typically you would want to have your homology arms somewhere between 500 base pairs to 1 kb. So a lot of labor has to go into cloning these homology arms on either side of your green fluorescent protein. The HDR also only occurs in dividing cells, and that's because it's operational in two phases of the cell cycle, both the S phase as well as G2. And so if you'd like to use knock-in technology in primary non-dividing cells or directly in tissue, this just simply won't work. The end result, however, is that you can get scarless integration. And so down at the bottom, I've schematicized a protein of interest that's been fused to green fluorescent protein. However, this is typically somewhere in the range of 1% to 0.1%, In some cases, with very small inserts, this can be as high as 5%. But the majority of the cases are the protein alone, which is schematicized next to it without the GFP fusion. In contrast, non-homology end joining is not limited to either the S or G2 phase of the cell cycle, and therefore it can operate in both dividing as well as non-dividing cells. In this case, I'm showing green fluorescent protein. You'll note that there are no arms of homology on either side, so this can just directly insert into your gene of interest. Somewhere between 30 to 40% of the time that you have the insertion, you can have scarless integration just like you have with HDR. The rest of the cases, you can have small insertions or deletions at the junction between your gene of interest and, in this case, green fluorescent protein. If these indels, insertions or deletions, if they're in frame, then you still have a functional fusion between, in this case, green fluorescent protein and your protein of interest. And this occurs the majority of the time. And so the advantage of using or leveraging non-homology end joining, one of the advantages is that you also enjoy a great gain in the efficiency and the number of percentage of cells that are effectively labeled. In the cases where there's a scarred insertion with an insertion or deletion that throws the subsequent GFP out of frame, or for instance, in epitope tag, those out-of-frame infusions are no longer detected as functional fluorescent protein or epitope tag. So essentially, those events are dark, and they're not part of the analysis in the subsequent functional analysis. Okay, so the next consideration to think about beyond the repair mechanism that you're going to utilize is the delivery mechanism. And so for CASTag biosciences, we use adeno-associated viruses, but there are other delivery mechanisms that can be utilized as well. So the first is electroporation. Electroporation, generating an electrical pulse to deliver your molecules of interest into the cell. Electroporation can occur with a high efficiency if the protocol for the electroporation has been optimized for the specific cell type that you're utilizing. The negative side of electroporation is that it can impact cell variability. So in many of these cases, at least 50% of the cells may be killed during the electroporation event. This is generally limited to in vitro cells in culture. This is where it's found most of its use. I would note that there are some cases where you can apply electroporation in Vivo. However, the majority of the cases are used in vitro. And of course, the biggest downside of electroporation is that it can require expensive equipmentation as well as extensive optimization for your cell type of interest. The next delivery mechanism are liposomes. And there are commercial kits for a variety of different types of liposomes, so they're quite easy to use. This also can have impacts to cell viability, depending on the cell type and the liposome that's used. It's also limited to in vitro delivery for the most part, and it's limited cell types that you can use liposomes to deliver your CRISPR reagents to. And of course, the efficiency is much lower than that of electroporation. And then finally, viral delivery is another option. And in this case, we utilize adeno-associated virus. The reason for this is that adeno-associated viruses, depending on the serotype that you're using, can have very broad tropism. So there's no optimization that's required. You essentially sprinkle the virus onto your cells of interest or inject it into your tissue of interest. And because of its broad tropism, it will infect these cells at a very high efficiency. So the transduction efficiency is typically very high. In vitro, this can approach 100%. It's gentle on the cell, so cell viability is very high. The only downside for adeno-associated viruses is that it requires some specialized knowledge for the viral purification in order to achieve high titers. However, this is something that is taken care of by CASTag biosystems when we deliver these reagents to your laboratory. So how does HiUGE work in the discovery, typical discovery workflow? So the first is come to us with a hypothesis. So you have your gene of interest, your protein of interest that you would like to follow up on. Contact our team of scientists. They'll help you select the locus and select the insertion site in order to make the fusion or the knock-in into your protein of interest. Our team then does the computational design of the guide RNAs and prepares those for you. They package these into high-titer adeno-associated viruses, deliver these to your laboratory where you then initiate your own experiment, either in vitro or in vivo. So here you typically are going to sprinkle the adeno-associated virus directly onto your samples or inject it directly into tissue. Incubate this anywhere from 7 to 10 days to let the virus transduct your cells for the incorporation of the fusion protein or the fusion into your protein of interest. And then follow on with the typical workflows that you have for data analysis and hopefully future breakthrough. So here's what the kits actually look like. So I just covered the services that we include. So that's the guide design and selection, the AAV packaging. These are pre-mixed for you so that all you have to do is take it out of these tubes and sprinkle it onto your cells or inject it into your tissue. Each kit comes with a guide RNA. It actually has 2 different guide RNAs that's pre-mixed with your donor of interest. So you tell us if you want how you want to label your protein of interest, we'll give you the appropriate donor. It comes with a positive control virus for a known and validated protein that works in your species of interest, about 50 microliters of high titer virus, and the titer here is 10 to the 13th. So this is quite a high purified titer that can be used even directly for in vivo use. And this 10 to the 13th, just remember that number when I get to the validation later in the talk. Okay, so that's some of the considerations and how this technology works. What are some of the applications that you might want to think about using this for? So here I'm going to cover 7 different examples of applications spanning everything from microscopy and Western blotting to protein truncation studies. I won't mention proteomics, although you can use this for proteomics since you're labeling your proteins of interest. So we have used this for immunoprecipitations, followed by mass spectrometry, and then also cell line creation. Okay, so the first example of an application is antibody-driven applications. And this is really what drove the development of HiUGE in my own laboratory, where we had novel proteins of interest, but the antibodies that we bought on the antibody market had lots of cross-reactivity and just simply didn't work. So in this case, in this example, we've incorporated an HA epitope tag that was directly inserted into, in this case, beta-3 tubulin. So that when endogenous beta-3 tubulin is expressed, it now has this HA epitope tag that we can use to detect it with HA antibody. Here's an example of sequencing across the junction site for the mouse beta-3 tubulin genetic locus. And you can see from the chromatograms a perfect insertion of a tag of that remaining part of the donor recognition sequence where this has been cleaved out of the payload vector, along with the HA payload that's been inserted into beta-3 tubulin. Now, I should mention that we've designed this epitope, our epitope tag kits, and we have several different epitopes, such that in either orientation that inserts into, it generates a functional fusion with the epitope of interest, and so it's in dual orientation. So following this insertion, here's a Western blot of the labeled neuron. In this case, these were primary neurons, the HA epitope tag versus negative control cells. And you can see that by Western blot analysis in the green that there's a single HA immunoreactive protein of the correct size for beta-3 tubulin, and the loading controls of GAP-DH are in red below that. Here's an example of what this actually looks like in the dish. And really the first time that we saw this, this really blew us away. So here you see in the green the immunostaining for HA immunoreactivity. And you can see a large number of neurons now that have been labeled for beta-3 tubulin. This works not only for epifluorescence imaging and confocal imaging, but also works for super resolution imaging. So it's multiple modalities of imaging that these epitope tags can be used for. And then the bottom example is stimulated depletion, excitation and depletion, or STED, super resolution imaging, where you can see individual fibers of tubulin and a dendrite from one of these labeled neurons. And in the red, you can see these beautiful synapses that are viewed as puncta along the dendritic shaft. So one of the advantages, because these payloads don't really, they're agnostic to the gene of interest, they'll insert into any gene of interest where we supply these double-stranded breaks, is that you can actually mix these several different epitopes into a single tube along with the targeting vector virus, and in a single experiment incubate, in this example, neurons with 3 different epitope tags so that you can rapidly optimize what type of fusion your protein tolerates and what type of, in this case, epitope antibody works best for immunostaining your cells or tissue of interest. So this is an example of cultured neurons where you can see with 3 different colors we've detected the 3 different epitope tags that we've simultaneously introduced into the, kind of mosaically into these neurons of interest. And here's an example where we've also done this by injecting it directly into Cas9 transgenic pups, let them grow up, and then section coronal sections through the brain, and you can see the kind of this beautiful labeling throughout the brain of 2 different epitope tags that we mix simultaneously. And I'll show some higher resolution images of that in just a minute. The next application, so that's for looking at cells either in culture or in tissue statically. What if you want to look at them dynamically? And so in this case, because of the high efficiency of non-homology end joining that HiUGE leverages, we can also, at high efficiencies, insert even larger inserts, such as fluorescent proteins. This is an example where we've used our dual vector system to insert in red fluorescent protein into a mitochondrial gene of interest to label the mitochondria and these primary neurons. And in just a second, you're going to see this movie play. It's going to loop through it 4 different times. Okay, so hopefully you saw there. You can easily image the dynamics of your proteins of interest. In this case, red fluorescent protein labeling mitochondria. The next example is DisrupTag. Here we've used this to look at the parts of beta-4 spectrin. This is a protein that's localized to the axonal initial segment of primary neurons. And here we're using structure-function analysis at the endogenous protein in order to determine which part of the protein is important for its localization. So here we've either epitope tagged it at the very C terminus with an HA epitope tag, or truncated it internally. Here are the results following that. So here you can see on the far end, the full-length tagged protein localizes tightly to these axonal initial segments. If we truncate in and we loop off this plexin homology domain, which is a membrane binding domain, the endogenous beta-4 spectrum is still localized at the axonal initial segment. If we target, however, the spectrum repeats internally for truncation, what we see now is that the protein becomes diffuse. So we can rapidly determine which of the protein domains are important for the localization of the endogenous protein as it exists in primary cells. And here's just a Western blot analysis showing the serial truncation in the 2 middle lanes of the truncated protein by Western blot analysis versus negative control. Okay, the other use that that we're quite excited about is using this to generate rapidly diverse types of cell lines. And so in this example, what we've done is we've labeled a protein involved in endocytosis called clathrin with GFP. So we've labeled the endogenous clathrin protein with GFP. We used HiUGE to label these cells in vitro. Then we fax sorted these cells, and within 2 weeks from the generation of the guide RNA and the cloning to the fax sorting, we were able to select for stable cell lines, encoding a functional GFP fusion protein with this endocytic protein clathrin. So this actually worked quite well for rapidly generating a wide variety of different labeled proteins within your cell types of interest stably. Another example is rerouting your proteins of interest so that they're no longer localized to where they normally are localized to. And this can be used for both gain of function as well as loss of function studies. So in this case, what we've done is we've taken an HA epitope tag and we've fused this to a nuclear localization signal. And we've inserted this into a cytoskeletal protein called ART2. Now, in primary neurons, ART2 is highly localized in these puncta, which correspond to dendritic spines at synapses. And you can see that in the top blown up sample. We've simultaneously incubated these neurons with a different version. So in the green version is just an HA tag, so you can see, or a MIC tag, so you can see where the endogenous protein is localized. In the red version, we've put in this HA nuclear localization signal. And here you can see now all of the signal is localized to the nucleus as the endogenous protein is dragged away from where it normally resides, which are in the green dots out in the synapses. Finally, the last application is for many proteins of interest, since you want to study them at the endogenous level. Many of these are expressed at very low levels. And so because of that, they can also be difficult to resolve based on imaging, even with the insertion of a single antigen epitope tag. And so here we've leveraged the ability to insert larger inserts. This is something called spaghetti monster. And here we've, so the spaghetti monster is essentially a dark version of GFP. And the spaghetti monster actually encodes within the framework of the dark version of GFP 12 different copies of the HA epitope tag. So this is a highly antigenic insertion so that you can easily detect even proteins that are expressed at very low levels. So here are some examples down below where we've used this to tag several novel proteins that are localized at inhibitory synaptic contacts in primary neurons. I won't go through the names of these proteins. It doesn't really matter, except that we're labeling these with this highly antigenic sequence and then verifying that they do localize to these types of inhibitory synapses using a positive control antibody that we know works. Okay, so those are some of the applications that you might want to use this novel knock-in technology for. Now I'd like to quickly go through some of the validation of these types of experiments. So the first question that you should always ask when using CRISPR-Cas9 based approaches are, what are the off-target effects? And so we've looked at this in multiple different ways. I'll show you data from one of them. So in this case, we've predicted the predicted off-target potential cleavage sites based on the guide RNAs for labeling our genes of interest. So, and based on these predicted off-target sites, we designed specific primers that would amplify the genetic loci of these off-target sites if our donor vector inserted into those off-target sites. And so in the top two panels from this Western blot, so here on the left-hand side in panel D, I'm showing the predicted off-target sites for, in this case, this was an example where we labeled a voltage-gated sodium channel with an HA epitope tag. In panel E, I'm showing Western blot analysis, where we looked at, basically this is a genotyping assay, genotyping for the off-target insertion. In the top panel in the middle panel are PCR reactions to look for either the forward or the reverse integration of this HA epitope tag. The first lane there is the on-target insertion. So you see that these primers very robustly detect the insertion that's correct. The rest of the The rest of the gel is showing an image staining for DNA if there were any off-target insertion sites. And here you can see that there are no off-target insertion sites at the predicted off-target sites. And down below, we've used these primers to PCR across the junction as a positive control, and you can see that all of these work. We've also done genome-wide analysis, looking for any possible off-target insertions. And what we have found is that these off-target events are very rare. So less than 3% of cells have any off-target insertion. And of the off-target insertions that we've been able to identify, all of these were in non-coding sequences. And so they weren't functional in-frame fusions. The other note that I'll make on off-target effects is that in labeling experiments for knock-in type experiments, each cell is an independent event. And so if your labeling is consistent across multiple different cell types, that's generally going to be the correct fusion. And so, in a way, you almost have a built-in experiment to verify independently multiple different events that the labeling is effective and robust. We've also looked at the efficiency of insertion by image analysis. In this case, we've targeted two different genes, beta-4 spectrin, as well as this sodium voltage-dependent sodium channel. And here we've looked at different viral titers and how different viral titers of the targeting and the guide RNA and the donor vector affect the efficiency. And you can see at the highest titer that we tested, this is 10 to the 11th, we get somewhere between, in the middle panels in B, you see that we get 29 to 34%, so around 30% insertion efficiency. When we supply these viruses to you, we supply them at 10 to the 13th. We've also looked at the ratio, so we've already optimized the ratio of the guide AAV, or the targeting vector AAV, with the payload AAV, and we find that the optimal ratio of those two viruses together is a 1-to-1, and so that's how we supply it in the kits that we send. Okay, so this works for a wide variety of diverse protein types. So this is just an image example of 12 different proteins that we designed guides for and labeled in primary neurons within a week. So this is quite easy to use and quite rapid. And we've overall, we've labeled cytoskeletal proteins, receptors, proteins that mark organelles. So you saw the example of the movie lighting up mitochondria in real time. Nucleus, membrane channels, including that voltage-gated sodium channel, signaling proteins, including kinases, proteins involved in vesicle trafficking, as well as, of course, unknown proteins, which are some of the more exciting ones. We've also validated this in vivo. So this, again, is an example of in vivo. So we also see this was an example where we injected the mixed donor as well as the targeting viruses together into adult mice, into the cortex, and labeled two different genes of interest that are localized to these axonal initial segments. So either beta-4 spectrin or this voltage-gated sodium channel. And you can see by marking the endogenous axonal initial segment with an antibody specific against beta-4 spectrin versus the HA-labeled structure, we can then calculate the efficiency of integration even in vivo. And again, we see around 20 to 30% efficiencies in vivo using these viruses. And again, not only can it be multiplexed in vitro to optimize the types of sequences that you might want to fuse with your protein of interest in vitro, but you can also do this in vivo. And so these are some of the pretty pictures from the multiplexed of two different antibody epitope tags, both HA as well as MYC, from a variety of different cortical and subcortical structures. And you can see this was for beta-3 tubulin, and so you can see that it's quite efficient and really gives a really beautiful labeling, mosaic labeling, even within tissue. So you can rapidly optimize your experiments in vivo as well. So in conclusion, I hope that you've learned something about this new technology and how powerful it can be, how easy it is to deploy. We hope that you'll contact CastTag Biosciences. We would love to hear from you so that we can help you guide, design guides for your genes of interest. We will send these prepackaged AAV for immediate use. We're also open to developing custom payloads. So if you don't see a payload or a sequence that you would like to insert into your gene of interest on our website, please contact our sales force or our technical support team. We would love to work with you to develop new payload vectors. Also, if you're interested in becoming a beta tester to test new payloads, we have several in-house that are currently being generated to empower in vivo imaging studies as well as proteomics. If you'd like to become a beta tester, again, please look at our website listed below, as well as contact our sales force or our technical support team. And with that, I'd like to thank you for your attention, and I'll take questions. Any questions that you have from this presentation.